In this video, we are going to explore one of the most fascinating parts of molecular biology: how DNA is actively metabolized, modified, and even moved around. This is part one of a two-part deep dive, and today we are focusing on the incredible enzymes that work on DNA and how they play a crucial role not only in nature but also in biotechnology. Now, if you've been following along, you'll remember that in our first video, we talked about the structure of DNA, how it's a double helix made of nucleotides, each with a sugar, a phosphate, and a nitrogenous base. And in the second video, we went over DNA replication, how that double helix unwinds and is copied. With the help of DNA polymerase, primers, Okazaki fragments, and more. So now that we know what DNA looks like and how it's copied, it's time to understand what happens next. Let's start by zooming in on a basic truth. DNA is not static. It doesn't just sit in the nucleus like a blueprint tucked in a drawer. It's constantly being replicated, repaired, recombined, and even reshuffled. And all of that dynamic activity is possible thanks to a class of powerful proteins called enzymes, specifically enzymes that metabolize DNA. So what are DNA metabolizing enzymes? These are enzymes that act directly on DNA to do things like cut it, repair it, extend it, unwind it, and even chemically modify it. Without these enzymes, our cells wouldn't be able to replicate DNA during cell division, repair it when it's damaged, or reshuffle it for genetic diversity. They're like tiny mechanics working constantly behind the scenes to keep the genome functioning. We can break these down into several main categories: restriction enzymes and DNA ligase and other DNA modifying enzymes like nucleases, helicases, and methyltransferases. Let's begin with restriction enzymes, also called restriction endonucleases. These were first discovered in bacteria, where they act like a defense system. Their job is to recognize specific DNA sequences and then cut the DNA backbone at or near those sites. These enzymes are named after the bacterial species they were discovered in. For example, EcoR1 comes from Escherichia coli, HIND3 from Haemophilus influenza. The naming is kind of code that tells you their bacterial origin. Now, restriction enzymes come in three types: type 1 and type 3. These are complex, multifunctional enzymes with both restriction and methylation activity. However, they don't cut DNA at the recognition site itself. Instead, the cleavage happens quite a distance away, sometimes over 1,000 base pairs. Type 1 enzymes like EcoK recognize asymmetric sequences and cleave far from their binding sites. Type 2, these are the rock stars of molecular biology. They recognize specific, often palindromic sequences, meaning the sequence reads the same forward and backward, and cut directly at or near the site. Because of their precision, they are widely used in labs for genetic engineering and cloning. Some restriction enzymes make sticky ends with overhangs that can base pair with complementary sequences. Others make blunt ends, which are clean, straight cuts. Sticky ends are especially useful in recombinant DNA technology because they help fragments stick together in predictable ways. Let's say we want to insert a gene into a plasmid vector. We use the restriction enzyme to cut both the plasmid and the gene fragment with the same sticky end producing enzyme. Then, when we mix them, the overhangs base pair with each other, and now they are ready to be sealed with ligase, which we'll talk about next. This technique is fundamental to genetic engineering, whether you are creating transgenic plants, producing insulin through bacteria, or assembling gene therapy vectors. Restriction enzymes are your starting point. Once DNA has been cut, how do we put it back together? Enter DNA ligase. This enzyme forms a covalent bond between a 3' hydroxyl group and a 5' phosphate group on adjacent nucleotides, essentially sealing the break. DNA ligase is critical in DNA replication, especially when sealing Okazaki fragments on the lagging strand. During replication, the leading strand is made continuously, but the lagging strand is made in small chunks. DNA ligase connects those fragments into one smooth strand. In biotechnology, ligase is indispensable for inserting DNA fragments into plasmids during cloning. It's used in everything from making genetically modified organisms to forensic DNA analysis. Here's a fun fact. Ligase is more efficient at sealing sticky ends than blunt ends, simply because the sticky overhangs help guide the pieces into place before the enzyme seals the bond. So if you are doing a cloning experiment in a lab, you'll often choose restriction enzymes that give sticky ends. Ligase activity is also influenced by temperature and cofactors like ATP. For example, T4 DNA ligase, derived from a bacteriophage, is often used in labs because it's robust and versatile, working efficiently at 16 degrees Celsius to promote hybridization of DNA fragments before ligation. Interestingly, DNA ligase can also join RNA and DNA, although it works more efficiently with DNA. Its ability to seal nicks, reconnect cut strands, and even recover circular plasmids makes it a multifunctional enzyme in molecular biology workflows. So you already know endonucleases cut within a DNA strand. Now let's meet exonucleases. These enzymes nibble away from the ends of DNA molecules. For example, exonuclease 1 degrades single-stranded DNA from the 3' end. Exonuclease 3 works on double-stranded DNA and removes nucleotides from the 3' end. It also has some endonuclease activity at aprunic sites, places where a base is missing. Val31 and exonuclease 7 can chew from both ends, making them useful for creating nested deletions. These enzymes are great for precisely editing or trimming DNA, especially when you need to prepare fragments for cloning or sequencing. Exonucleases are also used in techniques like exonuclease digestion mapping, where you determine the structure of DNA fragments by observing how they get degraded. They also play essential roles in cellular processes. For example, exonucleases are involved in proofreading during DNA replication, removing mismatched bases so they can be replaced with the correct ones. This helps maintain genetic fidelity, reducing the chances of mutations. Many of these exonucleases have preferences. Some only work on single-stranded DNA, others on double-stranded DNA. Some require specific ions like magnesium 2+ or manganese 2+ to function properly. Understanding these preferences allows scientists to use them strategically in experimental designs. Imagine your DNA as a tightly wound rope. If you want to read it or copy it, you got to unwind it first. That's the job of helicases, enzymes that unzip the two strands of the DNA double helix by breaking the hydrogen bonds between base pairs. Helicases are essential during replication, transcription, and DNA repair. Without them, the replication fork would stall, and no new DNA would be made. In fact, defects in helicase function are linked to serious genetic disorders, such as Werner syndrome and Bloom syndrome, which are both associated with premature aging and increased cancer risk. However, as helicases unwind DNA, they introduce supercoiling, like when you twist one end of a rope and it gets more tightly wound in another section. That's where topoisomerases come in. These enzymes cut one or both strands of DNA to relieve the tension and then religate them. There are two main types. Topoisomerase 1 cuts one strand, allows it to unwind, and then seals it back together. Topoisomerase 2 cuts both strands, passes another portion of the DNA helix through the break, and then reseals it. This is particularly useful in processes like mitosis. In bacteria, a special topoisomerase called DNA gyrase introduces negative supercoils, which helps maintain a relaxed state for replication and transcription. These enzymes are so important that they become targets for antibiotics and chemotherapy drugs. For example, ciprofloxacin inhibits bacterial DNA gyrase, while etoposide targets human topoisomerase 2 in cancer cells. Now let's talk about methyltransferases, enzymes that add methyl groups to specific nucleotides, usually cytosine, in DNA. In bacteria, DNA methylation is part of a defense mechanism. It helps them distinguish their own DNA from foreign DNA, which might be from a virus. If DNA isn't methylated at the correct sites, bacterial restriction enzymes will recognize it as foreign and chop it up. In eukaryotes, DNA methylation has more complex regulatory roles. It can silence genes by preventing transcription vectors from binding to the DNA. This is one way cells control which genes are active and which are turned off. There are two main classes of methyltransferases: maintenance methyltransferases, like DNMT1, which copy existing methylation patterns onto new DNA strands during replication. De novo methyltransferases, like DNMT3A and DNMT3B, which establish new methylation patterns, especially during early development. Methylation patterns are critical for processes like X chromosome inactivation, imprinting, and even aging. Abnormal methylation is often seen in cancer. For example, hypermethylation of tumor suppressor genes can silence them, allowing uncontrolled cell growth. These enzymes also play a role in epigenetics, study of heritable changes in gene expression that don't involve changes to the underlying DNA sequence. In other words, methylation helps cells remember which genes to turn on or off. So there you have it, an inside look at the core enzymes that keep our DNA in working order. From restriction enzymes that chop, to ligase that glue, to helicases that unwind, and topoisomerases that relieve tension, and finally methyltransferases that chemically modify genes. These proteins are the unsung heroes of molecular biology. Understanding these enzymes isn't just about knowing biology, it's about unlocking the tools we use in diagnostics, gene editing, pharmaceuticals, and personalized medicine. In part 2, we'll continue by exploring how DNA gets reshuffled and exchanged through processes like recombination, conjugation, transformation, and transduction. This is where things get really exciting, because it's how life evolves and how we've learned to engineer it in the lab. Thanks for sticking with me through this detailed dive into DNA enzymes. Make sure to check out the next episode for all things recombination. See you there.